The average human kidney is composed of roughly 1 million nephrons. 80% are referred to as cortical, while the other 20% are referred to as juxtamedullary, and they differ in three important ways. First, the glomeruli from cortical nephrons are located near the outermost part of the cortex, while the glomeruli from the juxtamedullary nephrons are located near the cortical medullary junction. Second, the loop of Henle from the juxtamedullary nephrons has an ascending thin limb, while the loop of Henle from the cortical nephrons lacks an ascending thin limb. And third, the efferent arteriole from the cortical nephron merge into the peritubular capillaries, while the efferent arteriole from the juxtamedullary nephrons merges into the vasa recta. Now each nephron is composed of a glomerulus, which filters the plasma, and a tubule, which processes what the glomerulus filters. Now once the filtrate or ultrafiltrate leaves the glomerulus, it enters the first part of the tubule, which is referred to as the proximal convoluted tubule because it has numerous bends. The proximal tubule is composed of three segments, the S1, S2, and S3 segments. The S3 segment is often referred to as the straight segment, and it is the only segment of the proximal tubule that is partially located in the medulla, while the rest of the proximal tubule is located in the cortex. The cells that make up the proximal tubule are cuboidal and have a prominent apical brush border with densely packed microvilli, which serves to increase the surface area for reabsorption and monitor changes in the ultrafiltrate flow. The numerous mitochondria are arranged in parallel with basolateral infoldings, which positions them optimally to deliver ATP to the basolateral sodium-potassium ATPase, which is responsible for establishing and maintaining the electrochemical gradient required for the reabsorption of the bulk of the ultrafiltrate along this segment. As the ultrafiltrate exits the proximal convoluted tubule, it enters the descending thin limb of the loop of Henle. The descending thin limb is located exclusively in the medulla, and its cells are characterized as thin, simple cuboidal cells, with a simple apical membrane that has very few and irregular shaped microvilli. In addition, there are a limited number of mitochondria and no basolateral infoldings, which suggests that the descending thin limb does not play a significant role in energy-dependent transport. However, it does play a major role in water reabsorption. As the ultrafiltrate exits the descending thin limb, it moves along the 180-degree turn prior to entering the ascending thin limb. Like the descending thin limb, the ascending thin limb is located exclusively in the medulla. The cells that make up the ascending thin limb are thin, simple cuboidal cells. They have only a few and irregular shaped microvilli and a limited number of mitochondria, which suggests that they too do not play a role in energy-dependent transport. However, they do play a role in sodium chloride reabsorption via paracellular pathways. Now once the ultrafiltrate exits the ascending thin limb, it enters the thick ascending limb of the loop of Henle. Now the thick ascending limb begins in the medulla and ends in the cortex as it passes between the afferent and efferent arterioles. The thick ascending limb cells are cuboidal, have an apical brush border with small and irregular shaped microvilli, a moderate number of mitochondria arranged in parallel with basolateral infoldings, which suggests that the thick ascending limb is involved in energy-dependent transport. In fact, the cells of the thick ascending limb actively transport sodium chloride and potassium via the sodium-potassium 2-chloride cotransporter, or NKCC2, which plays a major role in establishing the medullary interstitial osmotic gradient. As the ultrafiltrate exits the thick ascending limb, it enters the distal convoluted tubule, which is located near the outermost part of the cortex. The distal convoluted tubule cells are cuboidal, have a slightly convex apical membrane with few and irregular shaped microvilli, and numerous mitochondria that are arranged in parallel with the basolateral infoldings. They too play a role in generating the electrochemical gradient necessary for the reabsorption of sodium chloride via the sodium chloride cotransporter, or NCC. Now, as the ultrafiltrate exits the distal convoluted tubule, it enters the connecting segment. The latest research shows the connecting segment is composed of a mixture of collecting duct principal and intercalated cells, and some cells that share the ultrastructural features of both principal and intercalated cells, which suggests that the connecting segment functions more like the cortical collecting duct. As the ultrafiltrate leaves the connecting segment, it enters the collecting duct. We should mention that 4 to 5 connecting segments feed into a single collecting duct, which is divided into the cortical collecting duct, the outer medullary collecting duct, and the intermedullary collecting duct. Now, the collecting duct is composed of 2 main cell types, principal cells and intercalated cells. Principal cells are involved in salt and water reabsorption, while intercalated cells are involved in acid-base regulation. Intercalated cells are cuboidal with a convex apical membrane that lack microvilli. They have numerous mitochondria, which are distributed throughout the cytoplasm. They generate ATP, which is used to drive the activity of the hydrogen ATPase and the potassium hydrogen ATPase. Furthermore, intercalated cells are subdivided into alpha or type A and beta or type B intercalated cells. And finally, principal cells are also cuboidal with a convex apical membrane with a modest number of small microvilli and a moderate number of mitochondria distributed throughout the cytoplasm.